So this lecture is focused on the immune mechanisms that are involved with different types of hypersensitivity reactions and how those reactions are associated with allergic and different autoimmune syndromes. So the learning objectives for this lecture are: 1, to describe the basic immune responses and clinical examples that are associated with types 1, 2, 3, and 4 hypersensitivity reactions. And then to focus more on type 1 hypersensitivity reactions and to describe the major steps involved in the sensitization phase of allergic responses, and then to describe the major cells and soluble mediators involved in the effector phase of allergic responses. So as you know, immune cells are important, and they're important in not just protecting against infectious disease, but also in the pathogenesis and regulation of a whole variety of autoimmune and inflammatory diseases. Many of these diseases are quite familiar to us from personal experience or because they're widely described in the literature. Examples would include allergy, asthma, systemic lupus erythematosus, multiple sclerosis, type 1 diabetes, different types of inflammatory bowel disease. But also, I think it's important to recognize that immune responses and immune dysregulation are really associated with other very common clinical syndromes that we don't often think of as being associated with immune responses, including atherosclerosis and heart disease, cancer, obesity, and even Alzheimer's disease. And so just to give a big picture overview, hypersensitivity diseases are really defined as disorders that are caused by pathologic immune responses, meaning immune responses that cause tissue damage. And they can be associated with autoimmune syndromes, as I mentioned, but also they can be associated with just normal reactions against microbes. A lot of the tissue destruction that's associated with infection is not a direct effect of microbial damage of tissues, but an indirect effect of the immune response to that microbial infection. In addition, you may have immune responses that are reactions against environmental antigens, and that is typically what occurs during allergy. So there are 4 main classes of hypersensitivity responses: type 1, type 2, type 3, and type 4. And what this slide does is summarize the biological basis of those different hypersensitivity responses. And I'm going to go through this in a little bit of detail, so bear with me. But we'll review this again when we talk about specific diseases. So type 1 hypersensitivity responses are really dependent on IgE-dependent immune responses. And that's because IgE is critical for activating mast cells and mast cell degranulation. And examples of type 1 hypersensitivity reactions would be things like allergies, asthma, and rarely systemic anaphylaxis. Type 2 and type 3 hypersensitivity responses both involve and require IgG production. And what the difference is that type 2 involves IgG engaging with antigens that are associated with cells. And whereas type 3 involves IgG engaging with a soluble antigen and forming an immune complex that then deposits in tissues. But the end result for both of these immune responses is similar and reflects all the things that IgG interactions with antigen does. It induces phagocytosis, sometimes direct killing involved due to complement activation. Also, complement activation will result in the recruitment of other innate inflammatory cells like neutrophils and macrophages to the site of activation, and also which results often in tissue damage. Type 4 hypersensitivity responses, unlike type 1, 2, and 3, involve T cells as the critical cell that causes the immune pathology. So anything that has to do with a T cell immune response, T cell immune effector response causing tissue damage would be classified as a type 4 hypersensitivity response. And there can be a variety of examples of type 4 hypersensitivity responses, some of them that you're familiar with, including contact dermatitis. So if you have a nickel allergy or poison ivy, that would be an example of a type 4 hypersensitivity response. When we do skin testing for infection for TB, most of us get that skin test and we don't see a reaction. But if we did see a reaction, that again would be an example of a type 4 hypersensitivity response that's dependent on previously activated T cells specific for TB antigens. There can be aspects of type 4 hypersensitivity in certain types of chronic asthma and chronic allergic responses, because a lot of times these hypersensitivity responses may involve multiple arms of the immune response, so not just IgE, but also may involve Th2-mediated immune mechanisms that contribute to the tissue damage. So I'm going to go through all of these in a little bit more detail, but for the most part I'm going to focus largely on type 1 hypersensitivity responses. So type 1 hypersensitivity is also known as allergy or atopy, and it's really what disease that follows a response by the immune system to an otherwise innocuous antigen, so something that we wouldn't think of as being harmful. Another way of thinking about type 1 hypersensitivity responses is that they're immediate hypersensitivity responses. And what that means is that the reaction occurs within minutes or sometimes even within seconds after allergen challenge. It's by far the most common disorder of immunity and is estimated to affect greater than 20% of all individuals in Western countries. So type 1 hypersensitivity responses are all mediated by IgE, but they can manifest themselves in different ways, in part depending on how you encounter the antigen. So if the antigen is widespread or at a very high dose, you can have a systemic IgE-mediated allergic reactions that will manifest itself in the form of systemic anaphylaxis. If it's more localized in the skin, you can get acute urticaria or hives, also known as a wheal and flare response. Those of us who have seasonal allergies in the fall because of ragweed pollen, we're familiar with what seasonal rhinoconjunctivitis is like, and that involves kind of an inflammatory response of the upper respiratory tract and the eyes and the nasal mucosa. Some of us also are familiar with asthma, that is often an allergic inflammatory response associated with the lung, often triggered by common allergens. It involves lung inflammation and bronchial constriction and mucus production. And finally, many of us are also familiar with food allergies, so things that we eat through the GI tract also can induce an allergic response that's characterized by inflammation of the GI mucosal and may manifest itself in terms of vomiting, diarrhea, as well as itching and hives in the more upper GI tract. So for all the type 1 hypersensitivity responses, they all involve IgE, but it turns out that in order to get IgE production, you need to get T cell help to help the B cells undergo immunoglobulin isotype switching to produce the IgE molecule. And the major form of T cell help that's involved here is thought to involve Th2 effector cells. And we'll talk a little bit more about the role of Th2 effector cells in just a few minutes. But what characterizes allergic individuals is that they mount a Th2 immune response to allergens and produce high levels of IgE, whereas non-allergic individuals either don't respond at all to the allergen or will make other immunoglobulin isotypes, for example, IgG or IgM, and relatively little IgE. And so one of the key things in trying to understand why it is that people get allergies in the first place is why do people mount this type of immune response, this Th2 IgE dependent immune response? And the short answer is it's not entirely clear, but it has to do with the antigen characteristics of allergens themselves, individual genetic predisposition, and the type of or history of antigen exposure. So let's just talk a little bit about the characteristics of allergens themselves that make them prone to inducing this Th2 IgE effector response. So typically allergens are proteins, and that's because proteins are good at activating T cells. It turns out that allergens often have enzymatic activity and are proteases. That has a variety of effects that I'm not going to go into in detail now, but in part it allows them to get exposure to the immune system by bypassing epithelial cell barrier functions, and also by certain types of enzymes activate innate inflammatory cells to potentiate a Th2 type immune response. It also turns out that low doses of antigen also favors the activation of Th2 CD4 effector cells. Allergens are also typically of low molecular weight. Again, it allows them to easily bypass, or relatively easily bypass, barrier defense mechanisms because they are easily diffusible. Consistent with that, allergens usually have a high solubility and a high stability. And finally, because they're proteins and they come exogenously, those peptides need to be able to be presented by MHC class II, which is required for CD4 T cell priming. So let's go through a little bit of the steps that are involved with allergic sensitization. And so in this case, it's a cartoon illustrating what occurs when we're first exposed to ragweed pollen, for example. Usually we'd be exposed to this intranasally, and the allergens associated with the ragweed pollen would come in through the airway. Some of those allergens may diffuse through the epithelial cells of the airway and become internalized by resident innate immune cells that are in the extracellular space. Those proteins are digested into peptides and presented on MHC class II molecules. And they may migrate to a draining lymph node, or if you already have T cells that are primed to those antigens, the T cells will become activated and manifest their effector response. And this is really the key step in allergic sensitization, is initial priming of a T cell and allowing that T cell to differentiate into a Th2 effector cell. So once that happens, the Th2 effector cell will make interleukin-4, which feeds back on B cells, and interleukin-4 is a critical cytokine in promoting immunoglobulin isotype switching to the IgE isotypes. And so it promotes B cells to produce IgE that's specific for the allergen. And that allergen-specific IgE will then bind to high-affinity Fc receptors on mast cells and kind of prime those mast cells to be ready in case they re-encounter that allergen. So the mast cells are loaded with IgE, but the mast cells aren't activated yet. They're only activated upon subsequent exposure to the allergen. So this, again, is a critical part of the sensitization phase of an allergic response. But it usually does not result in an allergic reaction unless you're re-exposed to that allergen. So you have everything set up to respond really quickly in an immediate hypersensitivity way upon re-encounter with the allergen. But sensitization does not happen quickly. It happens over several days, and it actually may require several exposures to the allergen before you manifest an allergic reaction. Another example of allergic sensitization would be sensitization to a dust mite allergen. So again, dust mite allergen is one of the dust mite allergens anyway, is an enzyme called DRP1 that has enzymatic activity and actually can cleave adhesive interactions that are critical for barrier epithelial cells to stick together. And so because of that enzymatic reaction, the DRP1 is able to bypass the skin and get into the subcutaneous space where it can be taken up by resident antigen-presenting cells like dendritic cells. The dendritic cells then can migrate to a draining lymph node, prime a naive T cell, and under the right conditions, this naive T cell will then differentiate into a Th2 effector cell. And as I said, one of the characteristics of a Th2 effector cell is it makes interleukin-4, which promotes B cell immunoglobulin isotype switching to make IgE. That IgE then goes back into the and can bind to Fc receptors on mast cells in the skin, And so when you're re-exposed to that allergen, so this would be the sensitization phase up to here. So when you're re-exposed to that allergen, that causes cross-linking of the IgE receptor, mast cell degranulation, and that's when you induce the effector phase of the allergic response. So all these panels are just sensitization, whereas this panel here illustrates the effector phase that happens very, very rapidly after re-exposure to the allergen. So what happens when the IgE molecules are cross-linked when they're bound to the Fc-epsilon receptors on mast cells? And so I went through this a little bit in an earlier lecture, but just to review, you have IgE molecules bound, and then sometimes you have an allergen that has multiple epitopes, and those epitopes bind to the antigen binding portion of the IgE molecule, which is in turn bound to the Fc-epsilon receptor. And when those epitopes engage with the IgE molecule, they can cross-link the IgE molecules, resulting in cross-linking of the Fc-epsilon receptors. That signals the mast cell. And the immediate result of that signaling response is degranulation of lots of lysosomal granules that the mast cell has stored, and it releases their contents, which cause the immediate hypersensitivity response. The other thing that is illustrated on this slide is that when you have IgE bound to the Fc-epsilon receptors on mast cells, that also causes the mast cells to make more Fc-epsilon receptors to allow more IgE to bind and to increase or amplify an IgE-mediated immune response. And so let's talk a little bit about the mast cell and activation and the mediators that are released by mast cells, and this is true to a certain extent of basophils as well, although mast cells are thought to be the major tissue resident cell that mediates allergic responses. And so the thing that happens immediately within seconds or just a few minutes after cross-linking of the IgE receptor is degranulation. And degranulation results in release into the extracellular space of preformed bioactive molecules that act on the surrounding microenvironment to cause that allergic inflammatory response. And so within these granules are large amounts of histamine, serotonin, a variety of different enzymes that cause that immediate inflammatory response. A little bit later, these cells will also release lipid mediators or bioactive lipid molecules like leukotrienes or prostaglandins that are synthesized as a consequence of Fc-epsilon receptor signaling. This takes a little while, but still just a few minutes, and they're released into the microenvironment and also promote an allergic inflammatory response. Still later, hours later, because this requires gene transcription, translation, and protein secretion, mast cells also are a source of cytokines and chemokines that are secreted into the extracellular space that will help to amplify the allergic response and recruit other cells and act on the local endothelium to cause vasodilation. So then to summarize, the sequence of events that are involved with the effector phase of an immediate hypersensitivity response. The first thing is that the IgG that's already bound to Fc receptors on mast cells is cross-linked by re-exposure to allergen. And then mast cells are activated to secrete preformed granule contents. This causes the immediate symptoms. And then a little bit later, newly synthesized molecules, which is associated with a later phase of an allergic response. Some of these secreted molecules, for example, some of the chemokines, may facilitate recruitment of additional inflammatory cells like eosinophils, which are then activated also at that site to secrete enzymes and other bioactive mediators to amplify the inflammation. And finally, you have this cascade of inflammatory events that result in inflammation and tissue damage. And these are the clinical manifestations of this immediate hypersensitivity reaction or type 1 hypersensitivity reaction. I mentioned this earlier to a certain extent, and so if the reaction occurs in the GI tract, you'll have increased fluid secretion, peristalsis, that's associated with diarrhea and vomiting. If it occurs in the upper respiratory tract, in the eyes, in the airways, you'll have increased mucus production, sometimes bronchoconstriction, resulting in congestion and blockage of the airways associated with asthma symptoms, additional swelling of the airways, and general nasal congestion. Blood vessels, instead of vasoconstriction, blood vessels actually become dilated because of the actions of histamine. This increases fluid leakage into the tissues, increased flow of lymph to lymph nodes, increased cells and proteins in tissues, and increased effector responses and recruitment of additional molecules and cells to those tissues. Now, if this is systemic and you have lots and lots of systemic vasodilation, this can result in hypotension or low blood pressure, potentially leading to what's referred to as anaphylactic shock. And so how do you treat immediate hypersensitivity reactions? It basically involves targeting the different pathogenic arms of the effector response. You can target specific mediators, for example, antihistamines, leukotrienes, production or the biological action of the leukotrienes themselves. Beta blockers can decrease the bronchial constriction. You can also target general inflammatory responses that are more chronic by using steroids systemically. You can try and target the Th2 effector response by inducing other types of effector responses, for example, regulatory T cells or potentially Th1 effector responses that would also downregulate Th2 responses. This is thought to be the basis for why desensitization therapy by repeated injections of specific antigen is thought to ameliorate allergic symptoms in some individuals. You can also target the IgE molecule itself. There's actually, over the last few years, there's a commercially available antibody that engages with IgE and prevents the IgE molecule from binding to the FcεRI receptor on mast cells. And so there's a variety of approaches that are used like that. This is not by any means an exhaustive list, but this is the type of pharmacological approach that's used to treat allergic disease. So I'm going to move on now and talk about diseases that are associated with other hypersensitivity responses, so the type 2, type 3, and type 4 mediated diseases. Remember, type 2 and type 3 are mediated by antibodies, IgG antibodies. Type 2 by an antibody that recognizes cell surface or tissue-associated antigens. Whereas type 3 hypersensitivity diseases are associated with antibodies engaging with soluble antigen, which in turn forms an immune complex that deposits in tissues. And this is just a table, in type 4, hypersensitivity responses is mediated by T cells, and this is just a table illustrating some examples of autoimmune diseases that are associated with these different types of hypersensitivity. And I'm not going to go through and read them, you can read them yourselves. And so just to go through type 2 and type 3 hypersensitivity responses relatively quickly, they're both mediated by IgG antibody engaging with antigen. Type 2 again involves IgG engaging with tissue-associated antigen, where type 3 involves IgG engaging with soluble antigen, which then in turn deposits in tissues that are susceptible to immune complex deposition, for example, the skin or the glomerulus of the kidney. In either case, once you have the IgG antigen interaction, that induces a cascade of inflammatory results that's associated with antibodies, including complement activation, that will result in local tissue destruction and recruitment of additional inflammatory cells. So on this slide you can see the recruitment of neutrophils and macrophages to the site of complement activation. These cells become activated and release their own enzymes, and the combination of the antibody plus the recruitment of the neutrophils and the macrophages induces a cascade of inflammatory responses that can cause tissue damage. Type 4 hypersensitivity responses are mediated by T cells. This slide just gives a couple examples of those. One good example is type 1 dependent diabetes or insulin-dependent diabetes, where you have an immune response targeted very specifically to the beta cells within the islets of Langerhans and is thought to be primarily mediated by cytotoxic T cells. So that's illustrated on this slide, where you have cells within the islets of Langerhans cells. These cells make different hormones, but only the beta cells within the islets make insulin. And what happens during type 1 diabetes is you have very specific cytotoxic T effector cells that specifically target the beta cells because their T cell antigen receptor recognizes MHC class 1 peptide complexes that are presented by the beta cells that activates the cytotoxic T cell effector function that results in killing or lysis of the beta cells. And this is very targeted towards the cell that the cytotoxic T cell is interacting with and spares the neighboring cell. And so you have basically specific destruction of the beta cells of the islet, whereas sparing of the other hormone-producing cells in the islet. And so what causes people to develop these types of autoimmune diseases? And so the pathogenesis of autoimmune diseases is complex and probably involves an interaction of both genetic factors as well as environmental factors that cause a dysregulation of immune responses, resulting in an inappropriate tissue destruction. Normally, we have a whole variety of immune regulatory responses that we haven't really gone through in this series of lectures that are designed to limit the development of autoimmune diseases. But in susceptible individuals, these mechanisms are overcome and result in autoimmune tissue destruction. We generally think that autoimmune disease pathogenesis involves an interaction of multiple genes that are also influenced by environmental interactions. It's fairly clear that single gene mutations that are directly causally related to autoimmunity are very unusual, although it is also clear that there are certain genes that do increase the relative risk for developing autoimmune disease. The most common is the one that I mentioned previously in an earlier session. MHC haplotype, or the MHC genes you inherit from your parents, is the most common and usually the strongest genetic risk factor. The reason HLA is the dominant genetic factor is multifold. One is because of reasons that we've discussed previously, that the MHC haplotype or genes influence peptide presentation in the periphery, meaning that certain MHC molecules may be better at presenting certain autoantigens. The other reason that MHC molecules may predispose to the development of autoimmune disease is because of events that occur during T cell development in the thymus that we haven't discussed in this series of lectures. But it turns out that the development of the T cell antigen receptor repertoire is initially random, but then is selected to a certain extent during thymocyte development to remove T cells that have receptors that recognize self-antigens. And depending on your MHC haplotype, that process may be more or less efficient, meaning that you may have more leakage of T cells during thymocyte development that express receptors specific for self-antigens. And this is just a slide similar to one that I've shown previously, illustrating that there's a whole variety of different autoimmune diseases that are associated with different MHC or HLA haplotypes. In addition to MHC, which is encoded on chromosome 6 in people, there's a whole variety of other genes that have been identified by genome-wide association studies, and there are more and more genes identified this way every day that seem to be associated with an increased or sometimes decreased risk for developing autoimmune disease. But usually these associations aren't sufficient to confer a strong enough risk to be able to predict that somebody is definitely going to get an autoimmune disease, because there's a strong interaction of genetic predisposition with environmental factors that collaborate with each other to alter the risk of developing autoimmune disease pathology. And so this slide is just a summary of some of the environmental factors that are associated with autoimmune disease pathogenesis. I'm not going to go through this in a lot of detail, but just to quickly mention a couple of them. One would be, again, disruption of cell or tissue barriers to expose tissues or antigens to the immune system, whereas are not typically exposed in a way that will provoke an immune response. So a good example of this are so-called immune protected sites, like in the central nervous system, or in the eye socket, or in the reproductive tract. Another important mechanism that's involved with bypassing immunoregulatory controls is associated with infection in a variety of ways. And so when we're infected with microorganisms, that induces an inflammatory microenvironment. And those inflammatory mediators, those cytokines and other byproducts of the inflammatory immune response may cause a decrease in the threshold of activation, allowing autoreactive T cells and B cells to respond when they normally would not in the absence of that inflammatory microenvironment. And different variations on that overall theme are probably a critical factor in why some people get autoimmune diseases and other people do not. So for therapeutic approaches that are involved with treating autoimmune inflammatory diseases, we talked a little bit about this for allergies already, but I just wanted to circle back and talk about that a little bit more broadly. And basically, these therapeutic approaches are based on what we've learned about the regulation of immune responses in both animal studies and human studies, and involve a lot of the things that we've covered in this series of lectures. So it involves targeting co-stimulatory molecules that are important in regulating T and B cell activation, molecules like CD28, for example, or CD40, and other molecules that we haven't talked about. Other approaches involve targeting specific lymphocyte subsets, like targeting T regulatory cells, augmenting their activity, would potentially decrease a dangerous or destructive inflammatory response. Removing the antigen that's chronically deposited in different types of immune complex diseases. Specifically removing cells that may cause the immune response. So for example, depleting B cells using antibodies specific for B cells, or targeting cytokines that are associated with inflammatory responses, including cytokines that promote inflammation like tumor necrosis factor. One of the more widely known and pharmacological approaches to targeting autoimmune diseases that's been around for over a decade now is using antibodies or other approaches to target tumor necrosis factor. You have, in this case, you have an antibody to TNF, or in this case, you have a soluble form that's genetically engineered of the tumor necrosis factor receptor. In both cases, they will bind to the tumor necrosis factor cytokine and block its ability to engaging with receptors on tissues and block its ability to cause inflammatory tissue damage. And so obviously clinical studies have gone on to prove the efficacy of these types of approaches. And this is just an example from the textbook showing you the treatment with anti-TNF reagents alleviate symptomatic scores from patients with rheumatoid arthritis, and also decrease biomarkers of inflammation like C-reactive protein. And so there are many examples of biological modifier treatments that are like this, that are becoming approved more and more by the FDA. What's lacking, though, a lot of times are side-by-side comparisons of some of these newer mechanisms with older, more non-specific mechanisms in terms of whether or not they're really more efficacious or elicit fewer symptoms. And so that's something you should keep an eye out when you read about the next favorite drug of the day. And so that's the end of this session. I thank you for your attention. And please don't hesitate to contact me if you have any questions about this session or any of the other sessions.